Hello everyone and welcome to today's lab video. So today's video we're going to be going into tissue histology. So it's kind of like a redundant title, but histology is the study of tissues. So the focus of today, so what I did was I took a bunch of pictures of slides from the microscope, and now just to give you a heads up, I did take these pictures using my phone because the microscope I used to take these didn't have a computer attachment. So they're not perfect quality, but it gets the job done. And I didn't have every type of tissue available. I just chose the ones where I had the best slides I could find, and even so, not all of them are still great, but we can get a general representation of all the tissues. Remember the different types of tissues we have. We have our connective tissues, we have our cartilage, we have our bone, we have our nervous tissue, and so forth. So that all breaks down into a lot of different ones, though. So what I have here is just a general representation of most of them. And now what I would recommend you doing at home while watching this is try to draw some of your own and highlight the key features that I'm highlighting as I'm talking about this. I'm just showing it slide by slide. I thought about using my microscope and changing all the slides, but this would turn into an hour-long video if I did that. So here I just have it as a little presentation where I've already used a microscope and to get these images. All right, the first type of tissue are the epithelial tissue. I think I forgot to say epithelial tissue when I just summarized all of them. So we have the epithelial tissues first. Now, the epithelial tissues come in a bunch of different types. So I want to break that down first. So first, they can be either simple, which means they are a single layer, or they are stratified. So simple or stratified, meaning multiple layers. And then each one of these could then be squamous. They could be cuboidal, or they could be columnar. Oops. Now, I'm not going to write them. It'd be the same ones down here, but I'm not going to write them again. Another tissue type. So mainly, be able to draw out these little branch diagrams here. Another example that's not listed here is transitional. So that's one that can change shape between multiple different ones. So like in the bladder or the ureter, where there's a lot of stretch happening, could go from a cuboidal shape and pinch it down to a squamous shape. It could go from a columnar to a cuboidal shape. So remember, squamous is flattened, cuboidal is cube-like, and columnar is column-like. And another example of one that's not listed here, it's kind of in between, is pseudo-stratified. So it's usually pseudo-stratified columnar. So pseudo-stratified means it's false layered. So when you first look at it, you think it's layered, but it's actually not. All right, so now let's look at the different epithelia that we find. The first one here is stratified squamous. So stratified means this is a bunch of layers, and squamous means they are flattened. So stratified layers are named based upon the apical layer. So here I'm showing an example of the epidermis, which is the top layer on your skin. So down here is the dermal layer beneath this, and up above here is the epidermis end. So these down here, they start as cuboid-like cells, and up here they become flattened squamous. So a bunch of layers here, and the top cells are squamous, so it's called stratified squamous. So we find this on the skin, esophagus, and so forth, and different layers of levels of keratinization in each as well. So this is a simple one. This is just your skin. Next one here, simple cuboidal. So this is from the thyroid gland. So here in the thyroid gland, we see this row of simple cuboidal cells forming these little tubule type things here, little vesicles in a way. So here, when the thyroid proteins are being made, or thyroid hormones are being made, they, depending on how they travel through here and interact with iodine, we'll get to that in the endocrine system. But here, being able to move through, it's important to have this single cuboidal layer so that the thyroid properties or the hormones can move through this tissue without an issue. So this is a nice example of simple cuboidal. Now it's hard to find stratified cuboidal. It's a little bit more rare. So now here is a pseudo-stratified columnar epithelia. So pseudo is false layered. So when you look at this, it looks like there are different layers here. But this is just because the nuclei are a little off center. So some nuclei are down there, and they're not lined up in a row, whereas a typical layered, simple columnar epithelia, all the nuclei are lined up in a nice row. That's because in some of these, we have this might be a goblet cell right there, and that kind of pushes the nuclei out of the way. Another interesting thing about this one, because this is from the trachea, we see all these little cilia lining this as well, because this is like a mucus escalator helping to get debris out of the lungs. So this is, not only do we see the epithelial layer, but we also see an example of a ciliated columnar cell as well, which is can happen with these columnar cells. Also, they could have little microvilli, which are the little cellular extensions that come out as well. Next one then is ciliated, so have the cilia layer again. So there's the cilia. Simple columnar. So this one's not pseudostratified. Here we can kind of see these cells lined up in a row. And I know it's hard to see, but trust me on this one. These are column-like, not cuboidal. It's just if we went to a higher magnification, we could be able to see it a little better. But yes, here are the little cilia, and then here are the column-like epithelial cells. Now, this one is actually from the fallopian tube. So these cilia help move the egg down through the fallopian tube, like a little conveyor belt. Remember, cilia move back and forth, have a whip-like function, so they're moving mucus, eggs for this example, and so forth. And now here's that one I mentioned. This is transitional epithelia. This is from the ureter. So the ureter has a lot of stretch to it. And I know it's hard to see here and make out the different cell types because it's not the nuclei aren't differentially stained too well. But this tissue has a lot of stretch to it. So depending on how much fluid or how much urine is moving through here, if it's more stretch, it's going to take a columnar cell and push it down to something more like a cuboidal or possibly even a squamous cell, and it allows that stretch and recoil of these cells, which is kind of neat. So that's called transitional epithelia. Okay, so now we get to the connective tissue. So connective tissue is broken up into two main categories. Well, CT proper, cartilage, bone, and blood are the main groups. The first one here is CT proper. So CT proper can either be loose or dense. The loose ones could be areolar, could be reticular, or adipose, which is fat. So a lot of this depends on the type of cells or the type of fibers that make them up. And then and this is also a loose arrangement of each of these. Dense is then regular, where all the fibers are lined up in the same direction. Irregular, where they're all going different directions, like joint capsules. Regular is more like a tendon, where resist pull and tension in one direction, but if you hit it from the other direction, it could tear it. And then the last, the dense form here, is elastic, made up of a protein called elastin. So tissues that require a lot of stretch are made up of these. So this is CT proper. And then I might as well just draw it here too, the different types of cartilage. So we have the hyaline cartilage, we have the fibrocartilage, and we have elastic cartilage. So three types of cartilage. Hyaline is the one that has like the bluish tint to it, and it's the strongest. It's the one that makes up the costal cartilage of your rib cage, and the rings on your trachea are examples of hyaline cartilage. Fibrocartilage is one that resists compression and shock. So example of your pubic symphysis or your intervertebral discs, where a lot of compression between your back, your vertebrae happens. And then elastic cartilage, a good example of that. Well, there are two main examples. One is your ear. So you bend your ear, it's elastic. It's made up of elastin protein, so it gives it that little bit of stretch. And also your epiglottis, which is in your larynx and larynx, and then closes to block the trachea so food doesn't go down into your lungs. And then also part of the connective tissue, blood is considered a connective tissue, and bone is considered a connective tissue. Then after this, we'll talk about the three different types of skeletal muscle as well. But going back here to show a few examples of the ones I found in the classroom. And again, if we were in the classroom, we would be looking at the slides. You would be looking at all these in different microscopes and drawing them. You can actually see the light. So I took these images in the classroom, and there's a nice light on the ceiling that came up in the reflection there. So like I said, these aren't perfect images. These are cell phone images through the top of the microscope. Okay, back to the tissues here. This is CT proper, dense, regular. So there's an example of a tendon. So you can see these fibers running the same direction right here, and that's the uniqueness of this dense, regular connective tissue. Now this one is an example of dense elastic. So the elastic means there are these elastic fibers, and whenever you see these dark lines, like I'm drawing arrows to right here, these are elastic fibers. So these are from tissues that have a lot of stretch and recoil. Good example of this is the wall of the aorta. So coming out of the heart, it's a high pressure environment, 120 millimeters of mercury, which should be the highest. So that has a lot of stretch right there, every heartbeat. So we find out this is actually, a section from the aorta showing those elastic fibers. So this is a great example of that. Next one is a loose one. This is loose reticular. So reticular fibers are kind of what we find, they're a collagenous type of fiber, not collagen specifically. So they can form more branches, which allows other things to sit and rest on. They act like a scaffold and holding things together. So a good example of this is found in the lymph node. The fibers aren't differentially stained here, but you can kind of see them running through here and crisscrossing and branching. I know it is difficult to see, but then we have these darker pigment fibers, which are part of the immune system, white blood cells, B cells, and so forth, T cells sitting on these waiting for immune response, because these are from the lymph node, and the lymph node is an important organ in the immune system. So spleen, lymph node are two good examples of where we find reticular fibers. And think of them as a scaffold system. So this one we actually include two. First one is the cartilage. So this is the first cartilage we're talking about. Whenever we're trying to differentiate what we're looking at and knowing whether a tissue is cartilage or if a tissue is CT proper, you want to look for if it has these pockets. These little pockets are called lacunae. So here are the lacunae. Now, hyaline cartilage is the most dense form of the cartilage, so you should see a completely filled in ground substance outside. So inside here we have the chondrocytes producing the cartilage, so this is then surrounded by adipose tissue. But first here we see these pockets of the lacunae that have the chondrocytes in them. That's a key that you're looking at cartilage, chondro for cartilage. Outside here, so this kind of puts two together, we also have adipose tissue, which is part of the loose CT proper. So each one of these look like little droplets in oil, and that's what adipose tissue is. So there's an example of white fat here. And then here you can see the actual nucleus of these adipocytes, which are the fat cells for fat storage, energy production, and so forth. So this one is a good slide that shows two different tissues. Next up in the cartilage is the elastic cartilage, and elastin, we're staining differentially for that. Guess what we see? We see these dark fibers again. So if this was hyaline, you would not see any of these dark structures in here. But since this is elastic, we see these dark structures. So this could be from the epiglottis or the ear. I'm not exactly sure where this slide came from. It was just labeled elastic cartilage. And another thing we see here are the pockets of lacunae that have the chondrocytes in them. So kind of neat how that all works. Now, this is a horrible representation because it didn't have a good slide. It wasn't differentially stained for the collagen fibers, but I did still want to talk about it here. This is fibrocartilage. So this would be an example from the pubic symphysis or intervertebral discs. So here, these collagen fibers would be running the same direction, and let's say we get a compressive shock going this way, they push together then. I wanted to highlight what you can see here are these pockets of lacunae that do have those chondrocytes in them. So you can still make out some structure here. This is just a good example, and why I wanted to show it, of when you don't have a high resolution contrast stain, you can't see all the structures you really want to see. And now we get to the muscles. So we have three types of muscles we're going to talk about. So we have skeletal, or muscle tissue per se. We have cardiac, and we have smooth. So skeletal muscle, just some definitions here. Skeletal muscle is voluntary, meaning you control it. It is striated, meaning, so it's talking about the myofibrils and how these filaments line up over each other, these thick and thin filaments. It actually causes this like stripe-like pattern in the fibers. So voluntary and striated. Cardiac is involuntary. You don't have to tell your heart to beat, thankfully. So involuntary, it is striated. And it is also branched. So the heart beats as a unit. Calcium moves through it, and the whole thing, it's called functional synctium, and it works together. And that's because of this branching pattern, and so branched with these gap junctions called intercalated discs. So these are connections between these cardiac cells that allows the calcium to continue flowing through the whole system. Then smooth muscle, this is the lining of hollow organs like small intestine, stomach, and so forth. Smooth is involuntary and non-striated. So it doesn't have that striping pattern like we see in the other two. So it's important to understand these different representations and ways to define each type of muscle tissue. We'll get into more when we talk about each particular unit in that system, like skeletal muscles, which will be coming up later this semester. So here is a cross-section of skeletal muscle. So this is a little bit more difficult to see. So skeletal muscle can also be multinucleate, which means it has more than one nucleus. This is a cross-section. So imagine taking a muscle, turning it upright and cutting it this way. So you're looking straight down at the little fascicles here of muscle fiber. So the dark pigments here are the little nuclei. So here the myofibrils are actually facing up. So you don't see the striations in this. So I got another image here of a longitudinal section of skeletal muscle. This one also shows neuromuscular junction. So here you can see the myofibrils moving down through. So the last slide was a section like this, and then you were looking down from it from the top like that. So it is a little bit difficult to see, but there are striations here. You can see some light stripes. You have to zoom in pretty far and have a pretty decent magnification in order to see the striations here. But there are striations, and this is skeletal muscle. This is also shown the neuromuscular junction. So this would be an axon coming down here, and then this would be the synapse here that then controls this muscle. It's a motor unit. And then here's an example of smooth muscle. I know this one's hard to see as well, but there are some nuclei stained in here. And one confusing thing with this is you might first think this is dense regular connective tissue, but it's not. And if I were to examine you on this, I would say, what type of muscle is this? You would look at this and be like, okay, I don't see any striations going through these muscle fibers or anything like that. So I'd then say, oh, it might be involuntary too. So you'd be able to narrow it down and so forth. And then the cardiac muscle here. Cardiac muscle has branching, so we can see a little branch down here. It also has intercalated discs. So there's another little branch of it coming off. So right here we can see some of these intercalated discs. Might be hard to see on your monitor, but trust me, they are there. And those are the gap junctions that form between these different cells. And then we also, all the dark spots here are the nuclei. Most of these are just, mononucleate, have one nucleus, but some can be multinucleate as well. Depends. Okay, now another connective tissue. I should have put this one before the muscles. We have bone or osseous tissue. If you see a whole bunch of things that look like trees, that's osseous tissue or bones. In the center of each, we have a central canal where the blood vessels and veins are going. And then each of these little concentric rings here are little lamella. These little lamella are circling like this, and then we have other rings. We'll talk about these more when we talk about the microscopic anatomy of the bone when we get to the skeletal system. But these little black dots in here are where osteocytes are. Osteocytes are the bone-forming matrix cells inside compact bone. So this is an example of compact bone. I'm not showing an image of spongy bone here. We'll talk about that later when we get to the bone chapter, though. But this one's pretty easy to see. It looks like a bunch of trees. And then the other connective tissue here is blood. So blood, you're looking at cells that do not have a nucleus. So these are all anucleate. But we do see some purple pigments here. These are white blood cells. You don't have to know the different white blood cells now, but this is just a good representation of blood. If you see all these little spots that you don't see a stain in them, you don't see a nuclei in them, it's more than likely blood. I made a mistake one year of using a frog blood smear. Apparently frog blood is nucleated. So everyone was quite confused, even myself. So now we have a human blood smear slide, so we can see the anucleate red blood cells and the nucleated white blood cells. Some of these have a bilobed nucleus as well. So if you see a couple purple pigments and then a bunch of cells that don't have any stained nuclei, that's blood. And now the last one I have here, again, this wasn't every tissue example available. Like I didn't show a simple, I didn't show simple squamous because I couldn't find a good lung slide of the alveoli, which is a good example of where they come from, or a serous membrane. These are the important ones that we would have looked at in the laboratory, so I'm including them here. Then the last one here are the neurons. These are part of the nervous system, the main cells of the nervous system. There are other supporting cells in the nervous system, but here is just focus on the neurons. So right here would be the nuclei. Oops, come back here. The nucleus of this cell body of neuron. We see these little appendages coming out. These are called dendrites. And then there's also be a long axon coming out. Here is where the signal will go. They look like little spider-shaped cells almost. So these are called multipolar neurons, or different neurons come in different shapes and sizes, but we'll talk about that more when we get to the nervous system in A and P2. But here, just a general representation of the neurons for nervous system tissue. All right, so that's all I have for this chapter. If you have any questions, feel free to reach out to me. But if not, I hope you all have a great day, and bye-bye.